Hello, and welcome back to the Mechanochemistry Discussions. This seminar series is hosted by the NSF-funded Center for Mechanical Control of Chemistry, or the CMCC. I'm Ashley Martini. The goal of our seminar series is, as always, to bring the community together. Our seminars stream live on the third Thursday of every month at 11 a.m. Eastern Time. They are also available to watch anytime on the YouTube channel. I encourage you to check out the YouTube channel to view some of the excellent previous seminars we've had as part of this series, some of which are shown here. We also have a great group of speakers planned for the upcoming months, and we hope that you will join us for all of them. Before we get started, a few thank yous. First, Dr. James Batiste, the Center's Director, Jennifer Belsick, our Center's Administrative Coordinator, and finally, two CMCC students without whom the seminar wouldn't be possible, Katie Floyd and Ryan Kwok. Thank you ahead of time for joining us. Please do to continue following us and subscribe to the YouTube channel of CMCC Mechanochemistry Discussions to stay part of our seminar series going forward. Lastly, a few quick guidelines. Reminder that the seminar is being recorded. If you have questions during the seminar, you can either email them to us at cmccdiscussions@gmail.com, or you can post them directly into the YouTube channel. Either way, they will be propagated to the speaker at the end of the seminar to be answered at that time. Finally, with no further ado, it is my pleasure to welcome today's speaker, Dr. Rafael Bernardi. Dr. Bernardi got his bachelor's in physics at UEL in Brazil, followed by a master's from CBPF and a PhD from the Federal University of Rio de Janeiro. During the PhD work, he did an internship at the University of Pennsylvania, where he worked with Professor Michael Klein. In 2012, he joined the University of Illinois, advancing from postdoc to research scientist at the NIH Center for Macromolecular Modeling and Visualization, led by Professor Klaus Schulten. Since 2020, Dr. Bernardi has been an assistant professor at Auburn University and remains a co-PI at the Illinois Center, where he helps coordinate the development of the NAMD and VMD. It's my pleasure to welcome Dr. Bernardi to the Mechanochemistry Discussions.
Well, thank you very much for joining us here. Thank you for inviting me for presenting my work. I will talk to you today about the role of catch bonds in pathogenic bacterial infection. So these are very strong bonds that become stronger as you apply force on them, and I'm gonna try to explain to you how they are actually how these bacteria are using these bonds to cause very nasty type of infections, and how we can fight them, understanding how they work at the molecular level. So before I start reading to that, I'm going to show to you a little bit about what we developed here in my group. We have been working on doing what we have been calling in silico single molecule force spectroscopy. So single molecule force spectroscopy is an experimental technique where you can pull one molecule at a time for instance, in a biological world, and then you're going to try to understand how that molecule behaves under force. Okay? So in my group, we developed tools to do that in the computer, so we can work alongside the experiments and have an extra layer of details, because in the experiment, typically you don't go, you cannot understand, you cannot see as much as we can see in the computer, let's put it that way. So these are not, so the tools that we use are not very new, but they're quite recent. They were developed mostly in the 90s. So the computational setup that we use was developed both in Klaus Schulten lab and in the Group Miller's lab, Klaus Schulten University of Group Miller's in Germany, where you have a molecular dynamics simulation, which is basically we are simulating every atom of the system and see how that evolves over time. We add a spring on it, and then we pull on that spring. So we are basically mimicking how we would pull in the system, and as the spring extends, we can see how much force the system is applying on that spring. Because basically, if you put at a constant velocity, you can see the extension of that spring, and then you can measure what is the force based on that. And so the force will depend on how strong is the protein on the other side, how resilient to force the protein is. The same setup a little bit earlier was developed to do in experiments. So Hermann Gaub is one of the main pioneers. I put Hermann and Klaus here because we worked together on this for many years. And Hermann developed a way of doing that with biomolecules in the AFM, so in atomic force microscopy. So by doing that, what he does is he has this molecule of interest, like this interface, for instance, of interest, molecule 1, molecule 2 here. He can go down in his cantilever where one of these molecules is attached, and then he fishes for the other molecule that is on the surface, and when he pulls back up, he's going to measure what is the force that that molecule reacts with by seeing the deflection of this AFM. So basically how much this AFM cantilever bends, and that's something that he can measure. Okay, so as you can see, both systems are going to somewhat measure the same thing. You're going to have your molecule of interest, and you're going to pull on them, and you're going to measure the force that you have the reaction from that system as you pull. So at the end, what you have is a force extension curve. So you're going to measure what is the force. What is the force that the system has? The system receives by what is the force that you need to break or to, like for instance, to rupture a bond on that system, like you're seeing here, like the force, the peak force would be the force that you need to break the bond. Okay, so how much force is you see in biological world? So like what you have here is a bar that shows like how much force you have in experimental setup, pulling at the same velocity, the same idea. Most proteins are gonna generate force or react to forces under 100 piconewtons. So most of the biological world lives here, like it's just a couple of piconewtons most of the time. You have systems like strep avidin biotin that are like workhorses of biotechnology, and like a lot of people in molecular biology and biotechnology depends on that, and that's considered very strong, and it ranges from like 100 to 400 piconewtons, depending on patterning, depending on how you attach that molecule, or how you attach the strep avidin to your systems. And then you have the chemistry world, like the chemistry world starts at about 1.8 nanonewtons, so like 1,800 piconewtons, and goes much higher than that. So, okay, so you can clearly see biology lives here, chemistry lives here. One thing that we learned over these last few years, so that I started working with Gaub, and also at the time I was working with Paul Schulten, is that there are systems in between. So we discovered these cellulosomal systems, and those are very large protein complexes used by some bacteria that live in let's say, harsh environments, and they are actually very effective at breaking their food source or like cellulose. So the cellulosomes are like very large complexes. They are typically found in like hot springs, in bacteria that lives in hot springs, or in the rumen. And more recently, there was one that was discovered in the human gut. So we actually studied a few of them, and we were able to find quite a large range of force resilience in the cellulosomal proteins, making them stronger than strep avidin, but still far away from the covalent bond. But a couple of years ago, we actually discovered there is another class, a bacteria adhesin, that can withstand forces in the same range of the covalent bond without a covalent bond. So it's like biology replacing the role of chemistry in this sense here. So we actually studied all these systems for a long time. We have been studying them over these years, and I'm going to talk to you today mostly about our work on the bacterial adhesins, but we have worked on systems like below 100 piconewtons all the way to this nanonewton regime. So we have been trying to understand how mechanical force, how force resilience in proteins evolve, like how they evolved over time, and also how they work, like what is the mechanism the same? Is it something completely different every time? And let's go into this a little bit. Why bacterial adhesins are so important, so strong. So when you start having an infection by this bacteria, typically Staphylococcus bacteria, the most common ways you're going to have that is by having something large implanted to your body. It can be a contaminated medical implant, like a hip implant is a very common cause, a catheter, or sometimes like a tooth implant. All of that makes basically like a big hole in your body, and you have this material that is implanted to you, and there's a very good chance that that material can be contaminated. So these bacteria live in our skin; they are everywhere. So it's relatively common to find them. Like specifically when the Staphylococcus epidermidis, which is epidermidis, like it's in our skin. But when they are inside our body and they find the perfect condition, they can actually grow very fast. So when they start that, as soon as you have the implant, they're not going to bind to the implant itself, but the implant is going to be covered by like fluid from us, like that's the extracellular matrix. And as soon as that happens, this bacteria can start to attach the extracellular matrix. And what you're seeing here is a microscopy of that bacteria attaching. And you see each one of these spheres here is one of these bacteria. And as it progresses, it starts to form what we call biofilms. So they start to interact not only with the extracellular matrix, but they start to replicate, interact with each other, forming like a layer. So when you get an infection that got to this level, it becomes very hard to fight it with antibiotics and our medications that we use today. So can we stop it here? That is the idea that many people have been trying for a long time. Like we have tried vaccines in the past, like what can we do to stop it before? Well, the problem is that we don't really understand what happens here. What happens like in the beginning of the adhesin. That's the process that we have been studying. Okay, so when a bacteria, so like this is a general idea here, not exactly the bacteria that we're working. When the bacteria finds an abiotic surface, they're going to find this. The abiotic means like the material, like the hip implant. They're going to find a way of attaching to that surface, okay, by attaching to molecules that coat that surface. And then it's common to have some hydrodynamic force. It's common for it to have, like can be blood flow, can be urine if you're in the urinary tract, can be many different things, can be you coughing when it's in your lungs. What is going to happen is bacteria is going to have an adhesion force. They're going to have to bind strong to the surface until they start forming these biofilms. Okay, so then when they form the biofilm, it becomes harder. And during the infection, what happens is this biofilm forms, and then after some time, they're more mature. Some bacteria start to leave and start to form other biofilms, and the infection spreads out. Okay. So one thing that was proposed, and actually we have been working on, is that some of these mechanisms could be what we call a catch bond. And what a catch bond is basically if you look at the bond over time, let's say you're binding, you're bound to something, and then you let it just leave there. What is going to happen is after some time that bond is going to form, and it's going to form again, it's going to form again. That's what happens in any condition without force stress. Now, if you apply force, 99.9% of the bonds is going to be like, if you apply a force, try to rupture it, it's going to break. It's going to be easier to break than it is without applying the force. With catch bonds is the opposite. As you apply force, they become tighter and tighter. It's somewhat similar to like one of these finger traps. Like if you have one of those, it gets harder and harder to pull away as you pull on it. But if you know how to do it slowly, you can remove it. So like if you let it there, it's going to go away. But if you pull it, you're locked. So the idea is kind of the same. So this type of bonds, they are very good at keeping systems that are under mechanical stress. Okay, let's try to look at the system and see if they are these catch bonds, because this is one of the ideas, because if they're so strong, they could be one of the catch bonds, not necessarily. So we start looking at these adhesins. They have this huge family of adhesins, like some Staph aureus, some Staph epidermidis. In our group, we have been working with many of them, but like our first work was actually in epidermidis in the SDR here, the serine aspartate repeat protein. Specifically, we work on SDRG, and these molecules, so all of them, they bind to all kinds of things in our extracellular matrix, but these ones bind to FG beta, so there's fibrinogen on our extracellular matrix. Okay, so how they do it, they have this part here. So what you're seeing in blue, green, and red is part of the bacteria. What you see in orange is part of the human. So they have all these domains here called B domains, and then you have the A domain, the adhesin domain, that has this N2 and N3 group, and you have this orange that binds. So basically, it finds the protein finds the N terminal, so it finds the end of the protein from the humans, binds to it in a way that they call the dock, lock, and latch mechanism. So like it's open before. So this green part here is a different color because it's typically open when there's nothing bound. When it binds to the orange, it locks on top and closes this latch. And when that happens, the system becomes technically like more resilient to force. That is something that we learned. And the B domains were of unknown like role until a few years ago, when some of our collaborators showed that they should be working as shock absorbers. So like they break before the A domain breaks, basically. Okay, so now you can imagine that the bacteria can be on the floor, and that the whole thing, that protein is holding the whole bacteria. Of course, this is completely out of scale. The bacteria is way larger than the protein, but the idea is you have something large. It's under flow, and then you're going to have this, sometimes this one bone trying to hold it. So this bone needs to be mechanically stable. And sometimes you're going to have a few of them, but like it needs to be very stable to resist to that flow. This flow can be very large if you have an urinary tract infection. That's actually some of the most force resilient ones were found are from proteins found in the urinary tract. But like it varies, of course, depending on where the infection is, and that the necessity of this being very strong depends, of course, on where this infection is. So when we look at that, so this is my collaborator Lucas Miles from my Gallup lab. He's now at the University of Washington in Seattle. He looked at that in the experiments, and I was doing the same time in the computer. And the interesting thing here, like, the fun fact is, like, as soon as he came out with the results from the lab, I was visiting Germany at the time. He came to the office where I was, like, I think I broke the microscope, because the result was so much higher. The force was so much higher than what we expect. So, like, these are the fingerprints that we typically measure. Typically, the measurements that we see are, like, this is 500. So, like, if you look at strep avidin you would break around here. So we had this huge peak. So it's like completely unexpected. I had the simulations running, which I was not checking because I was working on something else. And I went to check the simulations, and this is my first result from the simulation. Also in the crazy high force. The mines are higher force because I pull faster than we pull in the experiment because of limitation of the computer, of course. And then we start doing replicas and more replicas, and we got to this. So this is the in vitro data, this is in silico data. We got thousands of simulations, and we could, if you see, they follow the same kind of pattern. Actually, if we fit them using what we call the Dudker-Hamazabo theory, we can see that we can fit the experimental data perfectly well using the simulations. These, by the way, are excluded from the fit here, so I'm using only the computational data, and I recover what happens at the velocities that they pull in the experimental side. So if I can do all of that, now I have a big advantage. Remember that I showed you that we get a force extension curve, both from the simulation and the experiment. You might think, like, so why do the experiment? Why do the computer, or why do the experiment? It's actually the cool thing is that the computers can now also give you where is every atom all the time, because we have the trajectory saved, and we have thousands of trajectories, so we can start analyzing that data. To put some perspective to you, the most recent work that we have been doing on this system that we are doing right now with some collaborators in Belgium, we generated about 100 terabytes of data in a month. So it's an enormous amount of trajectory data that we have to dig and analyze using, of course, all the AI tools that we have today. Like trying to find information inside, trying to understand what is happening. Okay, so there's still these 5 orders of magnitude gap in speed. We have also finding that we actually, in the last, like 2 years ago, we got a preprint on that. We're still working on improving. We want to get to the in vitro speed. We're still working on that. So we are trying to recover the result from the experiment, also in terms of velocity that we are pulling. We're trying to meet the two worlds in the middle. And that is something that has been done by Dr. Gomes, who's a postdoc in my group here. Okay, very strong. We all agree with that's very strong. Experiments and simulations agree with that. So why? The first thing that came to mind was when the structure of the system was solved by crystallography way before we did our work. They noticed that they point out that these phenylalanines would create what they call the bulge plug. Like there would be like this large thing, and you have a constriction. You can see here, there's a hole here, and you have this large thing. So if you have something large, very large, you pass through a small hole, you expect that that's what is going to make this strong. And then let's test that, because that's a very obvious hypothesis, easy to test. So we then and did the experiments in vitro and in silico. So this is the wild type, the 2 phenylalanines there. So we have 100% of force, let's consider that 100. To crystallize, they actually changed the peptide because it was easier to crystallize it with 3 phenylalanines. So we also tested that. So now we have 3 phenylalanines, which in silico gave us, of course, about 4% higher or 1% higher in the experiments. Then we start to cut it down. So let's remove one phenylalanine. Still have a decent range of force. Let's make 0 phenylalanine. Still have a decent range of force. And I'm sorry, I should stop that. So like you see, so that means that the phenylalanine is not important. The phenylalanine is not why. Like having this large thing is not why this is strong. It contributes, of course. You're seeing here, but you still have the same high level nanonewton regime, would still be as strong as a covalent bond at this level. So there must be something else. Then, of course, we tested 100 different things, and I'm going to tell you the one that worked. But if you look at this trajectory, so this is the trajectory from the simulation. I'm just showing one here for you. Of course, we have thousands, and we have to dig into, understand that well, and quantify. But this is when I'm pulling the system, and if I'm going to zoom in, so the orange part, remember, is human. The rest is from the bacteria. You're going to find this dashed kind of bonds here that I have. These are hydrogen bonds. And of course, these are beta sheets that are going to form hydrogen bonds. It's kind of a back-bond interaction. But you have to notice a few things. There are many of them, and they are in this kind of twisted shape. Okay, and like when it ruptures, they all break at the same time, and then it holds a little bit again, all break at the same time, and so on. So if I go back to that, I'm going to go back here, and I'm going to go to this slide that I showed you here. What you're seeing is that when you rupture the first time, you go down, you form a little bit some bonds, but then it's not as stable, and then you break again and break again. You don't see that in the experiment because our resolution here is way higher than what they have in the experiment, so we can see that better in the computer. But there is some evidence actually with more high resolution AFMs that they can also measure some of those disturbances. So if we summarize, then what we have there is we have these hydrogen bonds in this twisted shape, and this is like our quantification, which bond, who is binding to who, and what is the strength of that bond. And the interesting fact is like all back-bond interactions, and it's twisted. So why is the twisted so important? I've been talking about it here. Imagine the two ends are very flexible. So the two ends of these proteins are just like chains that are not in any specific secondary structure. Like they're just unfolded. So doesn't matter how the system binds to the human. Let's say I bind like this, and I'm going to start to pull this way. I have bonds protecting in all directions so that I can align the system before I actually get to the high force regime. By the time I get to the high force, I'm always in the perfect shear geometry. So I'm always in this geometry that I have all these hydrogen bonds happen at the same time. Because if I start in a weird position, I'm going to line again before, so that I cannot peel it, basically. I cannot peel this bond out. And this is how most of the systems unfold, like adhering that is strong, like tighten that is strong. They all are peeled off. They are not. They don't break all at once. Okay, so that would explain what happened here. Okay, so now the computers were able to explain this, but the computers can also tell you something that is very nice. If you're correct, we could go there and mutate every single amino acid to a glycine at this peptide. So like amino acid without the side chain, and we'll only have back-bone interaction that should be strong. And that's what we did. So this is our peptide that is only glycine. And as you can see, we are still in the nanonewton regime. Of course, it's not as strong as the wild type because the side chains are important. So there's this confinement here that you see, like the side chains kind of interact. But most of the resilience still come from only the backbone interaction. Okay, this is just the computer guy playing God, like with the computer, there's how can you test that? We were lucky because this system, there are many adhesins like I showed you before, and one of them, the CLFB, binds to this K10 as a keratin type of peptide that is basically a repeat of glycines and some serines sometimes. So there's basically no side chain. So like it's a very small side chain. And when we tested that, we actually observed that the system was also ultra strong. Okay, backbone interaction seems to be the most important thing. Now, if you think about it, this is an amazing evolutionary advantage for the bacteria because you can bind. So imagine, okay, bacteria is not thinking, but imagine the bacteria is going to bind. Let's bind to something else. You're the bacteria. If you bind, you're strong. What that means is that doesn't matter what you bind, If you bind, you're mechanically stable because the side chains are not important. So at that level, of course, for binding, the side chains are very important. For the affinity, the side chains are very important. And now is another thing. We need to decouple what affinity is from what mechanical stability is, which is something that most biochemists don't like are trained to not think like that. Are trained to think like the affinity is related to the K on, K off. But like under force, that's a new regime, especially when you're talking about the sketch bonds. They live longer as you apply, of course, the force, and the force is actually affecting that bond. So it's a different world. So, for instance, the affinity of these proteins, which are way stronger than strep-avidin biotin, is much lower than the ones of strep-avidin biotin. Okay, so that's amazing advantage. We want to study that too, but we were always curious about, is this really a catch bond? The problem is, to prove that it's a catch bond, you need to pull the system, hold at a force, and then show that as you increase the force, you're holding the system at that force and it's going to live longer. So imagine that you hold it under tension, it's going to break at some point. And as you apply higher forces, they should live longer and longer. Although the Gauss lab in Germany was amazing in terms of the capabilities, the AFMs would never be that perfect because they could hold it for hours. And at that level, you start having drift from the AFM. So basically, the AFM is not as stable, and the system would still be bound. So we could not measure that. Last 2 years ago, actually, almost 3 years ago, a group in Singapore was able to show, using magnetic tweezers, that this system was a catch bond. We were able to measure that at a much lower force, and we were able to show that the system could survive for hours under force. So now imagine this. So this is a system that binds and unbinds in seconds, maybe minutes, like normally. And now we are talking about under force, it can live for hours. Another perspective, most of these bacteria can replicate in hours. So it's so strong that the whole life cycle of the bacteria can happen while this is under tension, under force, there at high force. So now we have the experimental data from the work we did in 2018, with also the computation, and also the experimental data, the more recent from this group in Singapore. We decided to go back to our data that we had already, and let's try to understand this catch bond mechanism. So the idea here is the following, especially like looking at their experimental results compared to our results. When you're trying to combine, it's like you're trying to navigate through a free energy landscape. And let's say you want to go from A to B here. So this is in the Alps, close to Munich in Germany, where some of the experimental work was done. You want to go from A to B. Of course, any one of us here, look at that map, is going to try to find a valley and go through the valley. It's going to be much easier. And that's exactly what happens under thermal conditions, normal conditions, without applying forces. Now, if you apply low forces, very low forces, you might still be here. You might still be in this regime. But if you apply an extremely high force, you go to this crazy deterministic regime here that you're basically saying that you're going to just go straight. Doesn't matter what is in the way. Of course, that's a bit crazy. That's not exactly what you do. You're applying force. You're actually finding valleys that are new valleys, new accessible ways, because you have this extra force to go in there. It's like there's a roadblock here, or it's a huge traffic jam that you're not going to go through the easy path, but you find an alternative path that is not as easy typically. But because of that driving force that can be the traffic jam, you now go through that route. So that's basically what you're having in the system. So you're finding a different way of unbinding. We were actually able to measure all of that with our computational tools. We see that under the condition of the magnetic tweezer, the system is still somewhat flexible. This is a superposition of the system over time. While when we apply high forces, they become much more rigid. We could measure that rigidity. We could measure the contacts here and calculate the network of contacts and see that they are much stronger when you are applying higher forces. And we're able then to find the path that explains how force unbinds the system. That's something that we published last year. But it's still like, that's awesome. You're able to show mechanistically how that works. But we still have one question that we want to answer: how they became so strong. And that's something that have been puzzling us for a few years. And also in other systems, like we were able in the past to see in cellulosomes how some cellulosomes are weak, some are strong, and you can, when you just look at their secondary structure, you cannot see the difference. So you basically don't know why one is strong and the other one is weak. We have been studying that. We have been able to do some mutations and increase the force of some of them. We did that with coronavirus. We did that with the cellulosomes. Now you're trying to understand the system, which is, I would say, a little bit more complicated. So the way we are tackling that, and I don't have big results to show to you or anything like that, is we are trying to reconstruct ancestor proteins. We're trying to understand how they evolve to become where they are. And we have been doing that with bioinformatics and also doing experiments on getting bacteria that were isolated in the 1800s and try to sequence them and try to see their evolution, to see how the systems are becoming stronger or not, and also understand the variations in the mutations. For instance, we work with the veterinary school here to get hundreds of this bacteria isolates and try to see if mutations on those systems make them more or less mechanically stable and how they're evolving. So our idea is to try to understand how they evolve to become so strong, and are they still evolving in that direction, or are there evolutionary pressures making them going that way? So in summary, what I try to show to you today here is that first, the binding affinity is not necessarily related to the equilibrium. Like the equilibrium is not related to the mechanical stability. So like the pathway to bind and unbind can be very different under force or without the force. So you can have this catch bond system that have a completely different path. We can measure all this using single molecular force spectroscopy, both in vitro and in silico. We can use both the experimental and the computational side. They go hand in hand. It's not one is better than the other in any aspect. That's not the goal here. And also you have to remember that just because the computer doesn't mean it's cheap, like some of the simulations are way more expensive than experiments because you need supercomputers to do. So it's just that they are giving different perspectives to the same problem. So we can use all this trajectory analysis to understand how these bonds are activated in catch bonds. I also talked to you about how pathogenic bacteria causes like this catch bond to bind to the extracellular matrix, how we are using that to understand the binding pathway, and how we are able to predict these forces. With that, I just want to say thank you to, of course, the funding agencies like the NSF that has been supporting my work, and also the NIH, and also my collaborators, both at the University of Illinois, where I started like this work, and at the LMU in Munich, where Hermann Galb collaborated, also like Professor Michael Nash, who worked with me most of the cellulosome work in Switzerland, and Jan Liefeld, who's in the Netherlands. We work a lot on the SARS-CoV work, and at Bayer in Israel, who is like the person who discovered the cellulosome. So he's one of our collaborators on that. And most of the recent work on the bacteria adhesion is done with people in Louvain in Belgium. With that, thank you very much for your attention. I hope you have a chance to visit Auburn one day.
All right. Thank you very much for that presentation. We have a few questions from the audience. Let's start with this one. You show that force increases with loading rate. Can you tell us more about the physical mechanism behind this increase? Okay, so the necessary force to rupture a system as you pull on the system, it's basically, I can explain that in simple terms. So as you pull faster and faster, the force that you need to rupture is, I'm trying to find a simple way of explaining. So as you pull on the system, the higher the speed you pull, that's going to be associated with the strength of or the resilience of the system. So if you pull very slowly, you're basically doing your thermal condition. There's no force applied to the system. If you approach that to zero, and in that case, the system is going to rupture the same way it would rupture in normally in solution. As you add, as you start pulling faster and faster and faster, the force that the peak force that you rupture is going to be higher. If you imagine how that would happen in a real world, application, I can get this finger trap in the same way. As I pull, if I just keep it, it's stronger. At some point, it's going to break because I'm still pulling on it.
But if I just go faster and faster and faster, like what's going to happen is the force that I'm measuring is actually higher because that's like I'm applying this higher force immediately, and that's how I want to break the system. I hope that answers the question in simple ways. The other way would be a lot of math, that
All right,
equation that I showed before.
thank you. Here's another question related to pulling rate. It says,
Yeah.
first, that was a great talk. When evaluating the pulling rate effects on bonds, what is the experimental limit there for AFM or with magnetic beads? Is there an advantage for one approach over the other?
So, okay, so most of the literature is in silico, in single molecule for spectroscopy. It started with okay, this methodology started kind of at the same time, but a lot of the work was done in the AFM in the beginning. I think the technical issues were solved faster, maybe. So a lot of the speeds that you measure when you compare are from the range that the AFMs can do. So I think that's a common way why we worked on that system, on that level. So the advantage of each one of them is AFMs typically are much better at high forces. So to get the level of force that we have here, it's like 2 nanonewtons at that loading rate. Magnetic beads technically can do that, but your bead is going to be huge. Optical traps would not do that. I don't expect any optical trap to be able to hold it. Unfortunately, like an optical tweezer would not do it. So these techniques would have different ranges of that. They work better. Optical tweezers typically have a much higher resolution, but they work at low force. And magnetic beads I've seen work on different ranges, but they're not going to do the same. Another thing is most of the work that I see with magnetic beads typically doing force clamp. So you pull the system, hold at the speed, and then you calculate, hold at the force, and then you calculate what we call the dwell time. So you calculate how much the system is bound versus unbound. And the AFM, a lot of the work is done at pulling at constant velocity. So you're adding force, and then you measure the rupture constraint. There are different ways of using the same techniques. You can get slightly different answers from them.
All right, thank you. We'll switch gears a little bit here. What is the weakest part of the bacteria's latching mechanism? And is there a way to exploit that to keep the bacteria from binding to the medical implants, body, etc.?
So, so the way, okay, it's a complicated system in that way. Let me open this. The in the 2000s, there was a vaccine that got all the way to clinical trials to fight these adhesins. And the problem is at the time they did not know the mechanism like we know now, and that antibody for the vaccine was actually binding the other side, so it was not binding here close to the top here. So it worked really well in vitro because it's binding, but it actually never worked well in humans because it was not actually blocking completely the whole mechanism. Technically, the results that we're showing say that if you can make this latch here less stable, that would work. So you can imagine that you could do that by binding something large here that would make that more stable, or binding something here that would block it so it never closes. So these are approaches that we have been discussing. A common strategy of developing drugs, especially like in silico, is to find something that would bind in this position that would like kind of compete for the site. There is a obvious problem in doing that in this system. So there are two problems that I would say immediately. Number one is there are a bunch of adhesins like I showed. It's not just one type. Second is, for instance, this one here, this SDRG, it binds to the FG beta in the same place that the thrombin cleaves it to create this signaling peptide that makes blood clots. So if you add a bunch of these peptides or something that looks like these peptides, there's a very good chance you're going to die of a blood clot. So you have to play and balance, what you can actually do. So it's not that easy. There has been some ideas that you can do that for specific types of infections that would not cause big problems. Like you're not going to take a medication, but maybe something that is specific to bacteria, you can implant exactly in the place just there, and then you're not going to spread through your body. That be less dangerous. But still, probably the way to go would be to block the latch. That's my guess.
All right, let's wrap up with sort of a general one. Maybe it's a two-part general question. Do you think there will be any way to incorporate this strong binding mechanism into other technologies? And more generally, where do you see this work going in the next few years?
So yes, I think there are many ways you can use it. So like as soon as we published this work in 2018, people in single molecule world started to use this as a handle. So like every time you're doing those experiments, you need something that is strong, that you know, that is stronger than what you're studying with. Otherwise, it's harder. Like, so you can't do it. You can't study the system. So we don't know anything stronger. But if you want to study something in a simple way, you just have some handle that is strong that you can go there and fish for what you want, and then at some point the handle breaks. So this is a perfect handle, you know, because it's not going to break. So that's one of the things that I've been seeing people using. When we published the paper, there was a company, like an editorial kind of on our paper, that was talking about how this could be used as a super glue. And I've never thought about that, you know, to that day. And it's interesting because we don't think about that, you know, like clothes detergent uses enzymes from bacteria, like glues might use enzymes from or use proteins like this. So there might be many applications like that. I know of people trying to develop meshes that are ultra stable, you know, like for diagnostics, where you need like a chip and then you need this mesh that is relatively very stable and it's still a biological molecule, so you're not going to mess up with anything. All those things can lead to like technological advancements. Now on my research, what I think it's interesting, there are two sides of that. There is the side that is studying the evolution of this. I think it's a very interesting concept to be studied, like at the university level, you know, like how this evolved to be like this. Is this a common mechanism of the protein? So this is something that I'm interested in, and that's what NSF is supporting this work. There is another part of the work that we are targeting more like an NIH type of audience, that is, can we design things to bind like this? That's in everything. Can we design something that is going to fight this bacteria? Can we design things like this that are going to bind to cancer cells that we can use as a signaling for cancer cells? So can we bind, you know, can we use this in a different way? And that's about the direction that we are taking. So using this technology, using the computers to develop a better interface, to develop something that binds perfectly to different proteins now. And sometimes, okay, maybe fighting the bacteria, which is hard, but can also be to like I say, like diagnostics of cancer. You know, like you can bind to something else and then use that. You know, that's the kind of thing that we have been developing.
Wonderful. Thank you again for your presentation.
Thank you.
Thank you so much, Dr. Bernardi, for that outstanding presentation. And thank you again for joining us. Just a reminder that we've already had an outstanding group of seminar speakers as part of the series, and we hope that you will check them out on the YouTube channel. We also have a great group of speakers planned for the upcoming months, and we hope that you will join us for all of them. Thank you again.